and the polymerase active site. Here, single-stranded viral RNA is transcribed into an RNA-DNA double helix. Ribonuclease H breaks down the RNA. The polymerase then completes the remaining DNA strand to form a DNA double helix. Now, integrase goes into action. It cleaves a dinucleotide from each 3' end of the DNA, creating 2 sticky ends. Integrase then transfers the DNA into the cell nucleus and facilitates its integration into the host cell genome. The host cell genome now contains the genetic information of HIV.
So see how the enzyme was used to integrate, or integrase was used to add the viral DNA into the regular host DNA.
Activation of the cell induces transcription of proviral DNA into messenger RNA. The viral messenger RNA migrates into the cytoplasm, where building blocks for a new virus are synthesized. Some of them have to be processed by the viral protease.
So this is the process of translation, which we haven't really talked about yet, or haven't demonstrated by picking a video that I felt would be good to show you this. But this is how the viral proteins are being generated.
Protease cleaves longer proteins into smaller core proteins. This step is crucial to create an infectious virus. 2 viral RNA strands and the replication enzymes then come together and core proteins assemble around them, forming the capsid. This immature viral particle leaves the cell, acquiring a new envelope of host and viral proteins.
And the capsid is the part of a virus that is going to, it basically is a whole bunch of proteins that surround the genetic material of a virus.
The virus matures and becomes ready to infect other cells. HIV replicates billions of times per day, destroying the host's immune cells and eventually causing disease progression. Drugs which...
So that's just one example of a retrovirus that uses reverse transcriptase. And it also is a good model to kind of understand how viruses are replicated. So I think from here we'll move into talking about the process of translating messenger RNA into proteins. When translating messenger RNA takes place, there's particular transfer RNAs that are useful in bringing together the amino acids that are going to make up the protein. So a transfer RNA brings in amino acid, and an amino acid will form a peptide bond to another amino acid that has been coded in this process. And that entire process, when you have the development of this long sequence of chains of amino acids, is referred to as translation, with a protein being the output product. And so when messenger RNA is being translated, it's read. First off, the messenger RNA has to make its way through a ribosome complex, like we saw in the video. And then there are 3 codons, or 3 nucleotide bases that get read together. And when they're read together, they each get transcribed into a specific code that's referred to as a codon. So a codon is a 3 nucleotide base sequence that is read by another structure called an anticodon. So when the anticodon reads the codon, then that's going to signal which specific amino acid the transfer RNA molecule is supposed to bring to the ribosome to generate the protein. So one example of how this coding takes place is, let's think of a DNA template and the codes that are in that template. Okay, so we just have a standard guanine, another guanine, and another cytosine. When this particular code is transcribed into messenger RNA, guanine is replaced by cytosine. So cytosine is the code that we have if we have guanine. So we have two guanines, so that's cytosine, cytosine. And then if we have cytosine in the DNA template, then we code it with a G, which stands for guanine. So our DNA template is transcribed from DNA to messenger RNA by converting or changing out the guanines and coding them for cytosine, cytosine, and guanine. Now, because cytosine only pairs with guanine, the anticodon for this codon in the messenger RNA that's being read in the process of translation is going to be paired with an anticodon, and the anticodon is going to be guanine, guanine, cytosine. So see how C is being paired with guanine again, C is being paired with guanine, and then guanine will be paired with cytosine in the anticodon. Once this anticodon is translated, then you have the creation of the amino acid, which the anticodon sequence for this particular amino acid as read is going to be proline. And there's an entire chart that shows you how this kind of works. So here, when we look at the genetic code, you have UCAG at the top and then UCAG at the bottom. One thing I think I forgot to mention was the fact that in messenger RNA, you have the removal of a thymine group. Let me go back to an image. See, we had all these thymines. If this were messenger RNA, all those thymines would be replaced with what we call uracil. So that's why we're seeing in the messenger RNA code, or the codon chart, we see these U's being present. And that's because we use uracil in the place of thymine in this process. So as these codons are being read, no matter what order they are in, this could be AUG, it could be GUC, CCG, GGC, GGA, no matter what that is, whatever the anticodon tends to be is going to dictate which amino acids are being produced. Okay, so on here in this diagram we saw we had an anticodon of G, G, and C paired with a C, C, and G. Okay, so on the chart what we go to is look to see where we have that CCG being that codon that is being read. And so when we saw the CCG, we see that it reads for that of proline. Okay, so CCG gives us a proline amino acid group. So in the process of translation, when these codons are read, depending on what code is being generated, that will dictate which amino acid. So we can have serine, cysteine, you could have a stop codon, you could have a start codon, which typically is AUG. That's your start-stop codon. So that's where you know that where the translation process needs to start and to stop. Typically, AUG is going to be the start process. So, and oh, here it is. This AUG is the start process. UAG is the stop process. And AUG, the first amino acid coded for, typically is always methionine, which is what you would have right here. So this is just the code and how it's used to figure out which amino acid group is which amino acid is going to be generated based on the pair bonding that occurs between the codon and the anticodon. Okay, and then here is just an outline of translation as it's happening. So here's your messenger RNA that's moving through this ribosomal subunit. The messenger RNA is going through. The transfer RNAs is this whole structure right here. And your transfer RNAs are typically free-floating out in the cytoplasm. So they already have the anticodon affixed to them. So as these codes on the messenger RNA are being read, they will determine which transfer RNA will come into the ribosome. So like this first one, as it gets coded for, we have ACG, it looks like. Actually, I think this one would pair with AU, UA, and CG because that's they don't line up exactly as they should. But as these are paired together and this messenger RNA is being read, you have the proper amino acid being brought into the ribosome. And you see the way these arrows are moving, it shows you where the first location where this process happens. So you see you have the amino acid affixed to the transfer RNA that's free floating. So let's start with this example, CGG. So CGG is going to be pulled into this ribosome, and it's going to pair with its codon. And when it pairs, there will be a linkage that will take place between the amino acids. So these two transfer RNAs, one has valine, the other one has tyrosine on it. Both of them have already been coded properly in this ribosomal complex. So the anticodon, which is situated on the transfer RNA, is paired with the codon on the messenger RNA. As that's read, then if this is a match, these two amino acids are going to stay put. Eventually, the amino acids start to form peptide bonds between each other, and when that peptide bond is formed, then the transfer RNA is no longer needed. So the transfer RNA is going to leave the ribosomal complex and go back out into the system, or the cell system, and try to find more amino acids to transfer or to carry into the ribosomal complex. So that's why they're called transfer RNAs, because they transfer amino acids throughout this process. Okay, so hopefully this gives you a pretty great understanding of, what the central dogma theory is. I know it's a lot of information. This is a very long lecture. It's going to be one of the longer ones that we'll have all semester. And I know it's on very complicated material, especially with the molecular and biomolecule structures that we've been going over. So if you have any questions, please don't hesitate to email me, or you can also call me. Remember, I gave you guys my office number, 703-284-1555, if you have any questions about anything, or if you want further explanation on some of these processes. But overall, just make sure you get I'm hoping that you got a good understanding of what biochemistry is, the different biomolecules, which should really just be a review from your anatomy physiology courses that you've taken. And hopefully you understand now how our genetic material is used to convert messenger RNA into proteins. Okay, I'll talk to you guys later.